Hello everyone. I'm Salvador Roman from the University of Vigo. First, I would like to thank Richard Unsworth for his invitation to participate in this event about the World Seagrass Day at the Seagrass Project. My talk explains an article published in Marine Pollution Bulletin last year where we evaluated the short and long-term evolution of fishery-stressed Thostera meadows, which was the last part of my doctoral thesis. So, as we all know, seagrasses have suffered a general decline during the last 100 years, and several studies have pointed out to the anthropogenic pressure as the main driver of this negative dynamic. However, there are some good news because some recent studies have found a positive trend in the last years. And specifically here in the northwest of Spain, the seagrasses Thostera norte and Thostera marina form dense patches within the productive shellfish beds. But some conflicts have risen up between meadows conservationists and shellfishers. So understanding the historical meadow dynamics is crucial to resolve this conflict, where the high-resolution orthophotos could play an important role in this evaluation. So the main objective of this article was to understand the short and long-term changes of three intertidal meadows, and we proposed three hypotheses. First, the seasonal pattern will be the typical of temperate areas with a peak in late summer. Second, the long-term evolution will evidence different periods of losses and gains. And third, the meadow variability will be mostly driven by temperature and shellfishing effort. So as you can see, the improvement in the water quality wasn't in our initial hypothesis. So we studied the Thostera meadows present in three productive shellfish beds of the northwest of Spain that were from north to south, Testal at the Ria de Noia, Osarrido at Ria de Arousa, and Aseca at Ria de Pontevedra. And there, the meadows were mapped with a portable GPS of some metric precision in order to assess the short-term changes, and the historical orthophotos were photo-interpreted to investigate the long-term dynamics. To validate the use of these orthophotos, we compared the areas mapped by both methods after the growing season in 2020, and the coincidence was always above the 85%, so we validate the use of this method. In addition, three transits of 100 meters were done at the beginning and at the end of the growing season in 2020 and 2021, and at the end of that period in 2023. And there we defined the type of habitat found in each meter along this transit, and it was based on three categories that were recently harvested, Thostera norte, and bird sediment. Then we defined three potential drivers of variability that were temperature, shellfishing, and pollution. First, the sediment temperature was registered at the rhizosphere level between 2020 and 2023. And then the shellfishing pressure was evaluated as the number of harvested per bed and the harvesting effort in each specific area. Finally, the pollution was investigated with the temporal evolution in the construction of new treatment plants. Well, the first result about the short-term dynamics indicate a similar pattern in the three shellfish beds, because there were permanent meadows or patches in the mid and high intertidal areas, we see here, but more variable patches in the areas closer to the open sea, I mean in the low intertidal areas. And probably it was because the harvesting pressure was higher in these low intertidal zones, where, for example, here in Aseca, where the probability of the seagrass presence clearly decreased when the harvesting intensity was higher. So regarding the area covered by the seagrass, the three shellfish beds show peaks at late summer, as here, and minimas at late winter, okay, which was obviously related with the temperature that range between 15 and 20 during the growing season. And this seasonal variability was also evident in the data extracted from the transects here. Because, for example, in Testal, the probability of found recently harvested Zoster Norte or bar sediment areas was different in its sampling date, as we see here with these letters. And what about the long-term dynamics? So between 2004 and 2023, the meadows present within these productive shellfish beds in general show a clear pattern of progression. Also, there were some periods of losses, as we already expected. In summary, the shellfish beds showed a clear progression with 50% of gains in Testal and Osarrido and 70% of gains in Aseca. By contrast, here in red, we see the percentage of losses that were around 5% at the three sites. And why this positive long-term dynamic? Because we have seen that shell fishing affect the meadows expansion and that temperature are the typical of temperate areas. Well, this positive trend could be related with the construction of new treatment plants in the study area because most of them were constructed between 2000 and 2009, as we see here, and there was just 5 treatment plants before the 2000s. So the implementation of new and better treatment plants could have improved the water quality, which is crucial to maintain healthy meadows, okay, as we all know. So the best result of this study is the story of optimism that I'm sharing today because we demonstrated that conserve healthy meadows is possible even when the conditions are not ideal. Well, now I would like to finish this talk with some conclusions. In this slide, we can observe the main threats to the meadows conservation. However, my point here is to explain that while some factors are difficult to change in a short period of time, like global warming or sea level rise, there are other activities such as harvesting, water pollution, boating, or aquaculture where an effective local management effort could be enough to improve the environmental condition in just some years. So the key and feasible actions to protect the meadows will be first, reduce the physical impacts like boating, fishing, and dredging activities. Second, reduce the water pollution with more and better treatment plants to improve the water quality. And third, preserve the existing meadows and restore them when needed. Finally, I would like to say that the current situation of seagrasses is critical worldwide, but as we have demonstrated, local and positive actions can promote meadows expansion. And I hope that this negative trend turns into a positive one in the coming years. And that's all. Here is my email, just in case you want to contact me. And thank you for your attention. And finally, best wishes from our research team. And bye bye.
Hi, everyone. My name is John Lefchuk. I'm a scientist at the University of Maryland Center for Environmental Science here in the United States. First off, happy World Seagrass Day. I appreciate the opportunity to celebrate with all of you and share some of our work on the past successes and many futures of underwater grasses, including seagrasses, in the Chesapeake Bay, USA. For those who aren't familiar with the Chesapeake Bay, it's a large estuary situated on the east coast of the United States in the Midwestern Atlantic. And Chesapeake Bay is one of the largest estuaries in the world, meaning that it's a mix of freshwater from its many tributaries and saltwater from the Atlantic Ocean. Its watershed encompasses 64,000 square miles and covers 6 US states and the District of Columbia. and its enormous size has created some challenges for management, as we will see in just a bit, but fortunately ones that we've been able to overcome and lead to some of the successes that I'll describe. And the bay, as we call it, is historically notable as the first permanent English settlement in North America at the colony at Jamestown, which was established in 1607. Many of you may have heard of John Smith, pictured here, an early colonial leader and most famous for his role in the story of Pocahontas. Unlike in the Disney movie, however, Smith was a notorious liar, and historians believe that much of his interactions with Pocahontas were in fact made up. He was also hated by the other colonists after inciting a mutiny on the way to America, and for all intents, he was exiled from the colony shortly after their arrival. In 1608, Smith sailed around the bay and charted its lands and waters, searching for food and allies. Of the Chesapeake, he wrote, "Within is a country that may have the prerogative over the most pleasant places known, for large and pleasant navigable rivers, heaven and earth never agreed better to frame a place for man's habitation." In other words, a paradise on earth. In the same volume, Smith goes on to list the vast marine life he encountered on his voyages throughout the bay, including some mystery organisms called crevices and seconac, whatever those are. Underpinning all this amazing life was, and remains, underwater grasses, or what we call submersed aquatic vegetation, or SAV. SAV comprises about 14 species in the bay, of which 2 are true seagrasses, and the rest are brackish and freshwater plants. Curiously, Smith never wrote about SAV in any of his books, although he may have called it something different, so it's hard to say for sure. Not only does SAV provide habitat and food for fishes, crabs, birds, and mammals that he did write about, we now know it supports the region's valuable fisheries, protects shorelines, and captures and stores carbon dioxide from the atmosphere, helping us in the fight against climate change. In the 400 years since the founding of Jamestown, the human population living in and around the bay has continued to grow. As of the 1950s, nearly 13 million people called the watershed home. And as of 2019, this number has grown by 70% to 18.2 million inhabitants. And in the next 30 years, a further 4 million residents are expected to join the region. And along with all these people come the usual activities: urban and agricultural development, runoff, and pollution, all of which threaten the health and cleanliness of the bay and its surrounding watershed. These growing human impacts became so pervasive that in the late 1970s, the U.S. Environmental Protection Agency commissioned a report investigating the primary threats to the bay. The final report cited the loss of submersed aquatic vegetation and reduced water quality due to nutrient pollution, or what we call eutrophication, as high priorities for attention. At that time, the bay was already suffering from the effects of Tropical Storm Agnes, which in 1972 dumped torrents of rain onto the bay, changing its salinity so drastically that it eliminated half of all seagrasses in the region in a single event. So, to provide information on the status and response of SAV to management, and to other impacts like those from storms, the federal and state governments in the watershed ratified the Chesapeake Bay Agreement in 1983 to promote science and set targets for restoration in the bay, including specific actions to limit nutrient and other forms of pollution to promote recovery of the SAV resource. One major act of the agreement was to establish an annual SAV monitoring program beginning in 1984, which in each year since has photographed the entirety of the bay waters and its tributaries from the air. This imagery has been used to create a map of SAV in the bay and has allowed us to understand how it has changed in the intervening decades. Because of the valuable insight we've gained from this long-running survey, we can look at the total acreage of SAV in the bay and how it has changed since 1984. Despite some ups and downs, the long-term trend over time revealed a more than 200% increase in the extent of SAV as of 2017. So the major question being, why? Why have we seen such drastic recovery of this resource? To answer this question, we assembled a team of scientists and managers from universities and federal and state agencies in the region to analyze these mapping data alongside water quality data that has been measured concurrently since 1984. What we found is a cascade of impacts from land to water. In short, the conversion of land to agriculture leads to an increase in the use of fertilizer. The application of fertilizer leads to an excess of nitrogen, which in combination with increased flow from rain and other forms of runoff is delivered into the waterways of the Chesapeake. The presence of this nutrient pollution has a strong and negative impact on the underwater vegetation. But how? Once the nitrogen hits the water, it stimulates blooms of phytoplankton, or microalgae, that cloud the water and prevent light from reaching the bottom. As underwater plants need clear water to photosynthesize and grow, the resulting decrease in water clarity leads to a decline in SAV. So why then have we seen an increase in SAV over the last several decades? Well, efforts to clean up the bay, namely reducing the application of fertilizer and the resulting nutrient pollution, has led to a 24% decline in nitrogen in the water column. Less nitrogen means better water quality, which means more underwater grasses. As of that 2017 investigation, the bay had met its interim goal of 100,000 acres of SAV and was well on its way to its overall goal of 125,000 by 2025. Note that that's this year, so what's happened since? Well, it's not great news. Since that peak in 2017, SAV has declined in the bay and has yet to reach those same heights over the last 7 to 8 years. On top of continued, albeit lower nutrient pollution, other emerging impacts such as global warming have caused the loss of particular species of SAV, such as eelgrass in the lower bay. So, where do we go from here? To answer this, we must enter the multiverse. Led by my colleague Mark Hensel, now at the University of Florida, we use projected values of nutrient pollution, temperature increase, land use change, and other parameters in the next 40 years to suggest multiple future scenarios for the bay and to predict how SAV will react. We focused on 2 strategies: continued reductions in nutrient pollution, so staying the course, and the opposite, total deregulation. And then we simulated thousands of possible outcomes. Here you can see the past trajectories of SAV and these alternate universes playing out into the year 2060 under these 2 scenarios. What we learned from these simulations is that no action, or deregulation, leads to low but persistent SAV in the bay, here on the bottom in yellow. However, if we stay the course and continue to regulate nutrient inputs, we are expected to see up to 50,000 additional acres of SAV by the year 2060. However, we can see that even under future nutrient reductions, there are few scenarios in which we achieve the 125,000 acre target, again demonstrating that larger global phenomena such as climate change are having a heavy hand in determining the future of SAV in the bay. Ultimately, the story of Chesapeake Bay has been a successful one. Despite some recent setbacks, the bay is healthier than it has been in many, many decades. However, the message from my presentation today is clear. If we want to ensure the future of SAV in the bay, we must continue to regulate nutrient inputs and improve water clarity, or we stand to lose much of the progress we've attained in the last 50 years. You can help by getting engaged in efforts to clean up your waterways, by sharing these stories of success to show that we can do this, and by pressuring leadership to invest in managing valuable resources like SAV into the future.
Hi everyone, I'm Esther Thomsen. I'm a postdoc at GeoMar in Kiel, and today I will be discussing the threats of poor water quality to seagrass and its sources across the British Isles. As you'll see, this is a data set that was collected by many people over the past 10 years, and I'm super pleased to present this team effort today. In the British Isles, we have two seagrass species, Zostra marina and Nanozostra nolti, but this study only focuses on Zostra marina. Similar to the global trend, the seagrass loss in the last century has been catastrophic. Estimations are ranging between 50 and 90% of the seagrass area has been lost. One critical stressor, probably one of the main reasons for seagrass declines, globally is poor water quality and poor nutrient management on land and therefore input into the marine environment. The excess nutrients in the marine environment can cause phytoplankton blooms, excessive epiphyte growth, macroalgal overgrowth, all limiting the light availability for the seagrass, which can ultimately lead to a regime shift from a seagrass-dominated to an algal-dominated ecosystem. Colleagues from Project Seagrass assessed the seagrass health at a number of sites that you can see here in the map and correlated seagrass health to tissue nutrients in seagrass leaves. They found that seagrass tissue nitrogen content are about 75% higher than the global average. However, you can see here in the map that there are large gaps, especially in the north. So we have expanded the data set and have asked the following research question. What is the status of the seagrass meadows in the British Isles? Are the seagrass meadows under pressure from nutrients? What are the areas of concern, and are there any thresholds above which seagrass meadows are likely to decline? Over the past 12 years, we were able to collect seagrass tissue from 60 sites and also revisit some of the earlier sites. So the whole data set now comprises around 270 leaf samples from about 60 different sites. We measured nitrogen, carbon, and phosphorus content and nitrogen and carbon stable isotopes in the seagrass leaves. At about half of the sites, we were able to collect seagrass meadows characteristics as well, and today I will be mostly focusing on shoot density. On this map, you see the percent nitrogen in the eelgrass leaves shown in this color gradient with higher values in yellow and orange. You can see already that seagrass in the islands have lower nitrogen concentrations. So we divided the data into coastal morphological groups, estuarine, and if there's river runoff, and also if the meadow is more or less enclosed by land. Coastal is half water, half land, and islands are surrounded by sea. Here you see the groups on the x-axis and the total nitrogen concentration in the eelgrass leaves in percent on the y-axis. We found a gradient from estuarine to coastal to island with higher concentrations in the estuaries, indicating that the nutrients originate on land, and in the islands there might be a higher dilution factor. Comparing the values to the ZEN, so the Zostra experimental network dataset, which comprises samples from 50 sites across the Northern Hemisphere, you can see that mean value from the British Isles is higher than the average from the ZEN dataset. And if you are wondering what these outliers in the islands are, these are data points for Skoma Island in Wales, which is home to a large bird colony fertilizing the seagrass meadow. And this is quite typical for this data set, since there are so many different sites across such a big area. Every site has its particularities and different pressures as well. So we have higher values in the estuaries and higher values in general compared to other sites in the Northern Hemisphere. But how does this correlate to seagrass meadow health? Seagrass shoot density is often used as an indicator for seagrass health, with high densities being considered healthy. Here you see the shoot density plotted against the percent nitrogen in the seagrass leaves, and there's a significant negative correlation between the two parameters, indicating lower seagrass health at high nitrogen concentrations. The model explains only 42% of the variation, but as I mentioned before, the sites all have their own particularities and potential other pressures as well. So for this data set, this is actually quite good. Plotting the mean values from the Zostra experimental network, that is at 470 shoots per square meter and 2% nitrogen, you can see that only 3 meadows in the British Isles have similar densities. And only island and coastal samples have lower nitrogen concentrations in the leaves. Looking at the data, I would like to hypothesize a nitrogen threshold above which seagrass is likely to decline. Actually, above 3% nitrogen, the shoot densities are all quite low, only about 125 shoots per square meter and lower. But what is the effect of the anthropogenic nitrogen on meadow health? Is there actually a measurable long-term effect? The long-term data in the UK is unfortunately very scarce. However, my colleague Chiara Bertelli and team from Swansea University and Project Seagrass have previously investigated trends in a few meadows. So let's have a look at them. And in this data set, we find 2 island sites, 1 estuarine site and a coastal site. And the team has looked at shoot density over time. So let's start with the first island site, which is the Isles of Scilly. Here and on top on this gradient you can see the percent nitrogen in the leaves. The Isles of Scilly have a relatively low leaf nitrogen content, and there seems to be a slight decline in seagrass density. However, there's another big pressure at the site, which is physical damage from anchors, and as there's a lot of boating and sailing activities going on in the Isles. The second site is Porthencline, a coastal meadow with higher total leaf nitrogen, and this site has considerably lower shoot densities. It is located right next to a large golf course that probably has an impact on the nitrogen content in the leaf. And there's also quite a lot of boat activity going on. Next is an estuarine site, Little Wick, which is influenced by all sorts of industry and agriculture as well as settlement. The total nitrogen concentrations in the leaves is higher than 3% on average, and you see a strong decline in seagrass density in the last 20 years. The second island site is Skoma Island that I have mentioned before. It's a seabird island with consistently high nitrogen values in the leaves and quite low shoot densities. So excess nutrients leads to thinning out of the seagrass meadow. But what could be the mechanism behind this? Here you can see the CN ratio, so the proportion of nitrogen to carbon in the seagrass leaves. The CN value can be an indicator for seagrass light availability, with values below 20 indicating light limitation and values below 15 indicating severe light limitation. Again, we find a gradient from the estuaries to the coastal sites to the islands, which was to be expected since estuaries can naturally be quite turbid. But we did not expect that 55% of the samples from the British Isles actually indicate light limitation. You can approximate the source of the nitrogen if you look at the delta 15N values, so the isotopic composition of the nitrogen in the seagrass leaves. Low delta 15N values, for example here in the Isles of Scilly, indicate that the nitrogen stems from atmospheric deposition. In Porthencline, here the little golfer, the higher delta 15N values indicate, for example, the fertilizer from the golf course and sewage runoff. In Skoma Island, we are seeing the influence of the guano from the bird colony, and the highest values we found in the estuarine site in Littlewick, indicating input from sewage and manure. And this brings me to the elephant in the room, the enormous amount of sewage spills that are discharged legally and illegally every year in the UK, and the state of the insufficient sewer system, which is a huge problem and leads to seagrass declines, together probably with runoff from agriculture. To conclude, Zostera marina in the British Isles is at risk from poor water quality, especially in the estuaries. One major threat is raw sewage and agriculture, and the quantification of nutrients and stable isotopes in seagrass leaves can be used as a quick and reliable assessment of potential nutrient pressures. At leaf nitrogen concentrations above 3%, seagrass meadows are at risk from excess nutrients. As a next step, we want to correlate the water nitrogen concentrations to seagrass leaf nitrogen content to understand if there's a critical value that can be fed into legislation like the Water Framework Directive. This will also fill the gap if we want to assess the health of a site without seagrass, and this then can also inform restoration projects. Then we should also challenge the threshold of 3%. So if anyone else is working with Zostera marina, please check your seagrass. And we desperately need to create long-term monitoring data that triggers action, and we should relieve pressures because this can lead to seagrass. I would like to thank a bunch of people who all helped with this project, and also quite a lot of funders, because this has been quite a big project over the last 12 years. And if you're interested, here's some literature, but also you can get in touch and ask for more.
Hello, my name is Dr. Chiara Bertelli, and I was really happy to be asked to give a talk for this webinar in honour of World Seagrass Day. My presentation today is going to cover some of the work I've been involved with on the RESO project, which stands for Restoring Seagrass for Ocean Wealth. I'm going to be talking to you about habitat suitability models and what transferability of them actually tells us about the potential restoration sites and how they differ, possibly due to water quality in places. To go anywhere near achieving global targets for net zero emissions, the protection and the restoration of our existing natural resources is required. Complacency is not an option, but active and targeted restoration is needed to recover what has been lost, but also to enhance environments as nature-based solutions for tackling climate change. Coastal habitats, including seagrass meadows, have been identified as key contributors to marine nature-based solutions. If you're not familiar with habitat suitability modelling or species distribution modelling, it is a method that relates biodiversity or species observations at locations to the prevailing environmental conditions. It uses algorithms to explain the biodiversity-environmental relationships based on niche theory, whereby different species fill niches within an ecological community based on the conditions it needs to survive. This enables predictions of species presence and distribution in space and time. These models are used for a number of purposes, such as species reintroduction, species management and restoration projects, and can be used to forecast the likely responses of these species to predicted future changes to conditions from climate change. Restoration of coastal habitats around the UK has rapidly grown in recent years, particularly for native oyster reefs, for seagrass meadows and for salt marshes. Around the UK, Zostera marina, the largest subtidal meadow forming species that we have, was once really extensive, but large swathes of this have been lost since the 1930s, significantly due to water quality issues. There is therefore a great potential for seagrass restoration around our coast, but only if conditions are suitable and if water quality issues have been addressed. The most important environmental condition that seagrasses as plants need for growth is light, and light needs to be able to penetrate down through the water column. This means that in temperate waters like ours, depth or bathymetry is often a limiting factor. In pristine waters, Zostera marina can grow down to about 20 metres, but in turbid waters, depth can be limited to more like 3 metres. This is why water quality is so critical for seagrasses to be able to thrive and to be considered when restoration sites are chosen. So how does habitat suitability modelling work? This diagram shows the flow of habitat suitability or species distribution modelling and what is involved at each stage, specific to the models that I have been producing for the British Isles. Species data is collected and it needs to be curated. You need to have good, accurate presence data. For Zostera marina around the British Isles, there are a range of sources that can be used and compiled from official monitoring survey data, but also citizen science observations such as from Seagrass Spotter. If absence data is available, this will also help the modelling process, but if not, then pseudo-absence data can also be produced. Environmental predictor variable data is really needed that spatially covers the presence points for the area that you're looking at to model. These will be chosen based upon ecological knowledge of the species you are looking at. Environmental data of the best resolution available should be chosen. Layers are standardised and clipped, so all of the same range and resolution is used. Collinearity tests are performed at this point to identify collinear variables that can then be removed at this point. From here, the models are fitted using a choice of either a single model type, such as GLM, or random forest, or an ensemble of methods can be used, which allows a range of algorithms. And then this takes a weighted average of all the model types and all the model runs that you do. Models are evaluated using a range of methods that enable you to cross-validate your data and to enable you to visualize the data to see how it fits against observed data. Final outputs are often visualized as maps showing the range of suitability calculated by the model as its habitat suitability, which is a probability from 0 to 1. For the RESO project, a number of case study sites were chosen around the UK based upon environmental gradients, but as well as restoration effort that was being carried out there and also community engagement levels. The sites included small seagrass meadows on open coast to sites which included lagoons, estuaries and whole island archipelagos. Ensemble models were produced for the entire British Isles and Channel Islands where we had good species presence data. The same model method and environmental data was then used to create smaller models for each of the case study sites so that results could be comparable. These are the results for the models that were created for the case study sites that were chosen for the RESO project. All of the models performed well based upon the model metrics that were calculated. There were a few issues and limitations to models where the environmental data did not quite reach the presence data which happened in one of the areas, such as Craigneish, and where presence data was limited or even clustered, such as in Porthincline. This meant that some of the model runs did not converge, but as a whole, most of them did for all of the algorithms that were performed. And so as an ensemble, all of the model runs worked well enough for us to produce these maps. So these case study sites show a predicted probability of suitable environments for seagrass to grow, where the dark red shows closer to 1 and the blue shows closer to 0. So this graph shows the variable importance for each of the sites and also for the British Isles model from all the ensemble models produced. The variable importance is calculated using two permutation-based methods, a correlation-based and AUC-based. Here I'm just presenting the correlation metric, which had very similar results to the AUC metric. As you can see, there are vast differences between the variables that are explaining seagrass suitability between sites. For Orkney, PAR, or photosynthetic active radiation, is the most important variable. But for Milford Haven, Studland Bay, Craigneish and the British Isles models, bathymetry is the most important predictor variable. This is not unusual, as we know that light and depth are going to affect the amount of light, enabling seagrass to photosynthesize and to grow. But we can see in some local areas other variables are having more effect on predicting suitability, such as salinity in the solent, for example. For restoration purposes, transferability would allow suitable habitat to be identified more reliably in areas where species perhaps have been lost and are no longer present, or areas where the absence of surveys has led to a lack of species presence data, yet these areas could still potentially host seagrass or be suitable for seagrass restoration. Models that are generalised enough would have the ability to be extrapolated or to be transferred to other scenarios. Previous studies have found good transferability for algorithms such as GLM. Therefore, to further explore and compare the variable influence on seagrass presence and the transferability of habitat suitability models between site areas, quadratic GLMs were chosen and AUC metric was used for comparison and for performance. The UK model successfully extrapolated to many other sites with excellent AUC scores greater than 0.1, as you can see in this figure on the right. This was excellent for Studland Bay, for the Isles of Scilly and for Orkney. There were good scores for Porthincline and Milford Haven, showed by values that were greater than 0.7. The GLM for Studland Bay had good AUC scores when applied to Porthincline, Orkney and the UK. So if you have a look here, when it was applied to Porthincline, Orkney and the UK, these scores were actually quite high. The other good score was when the Orkney model here was applied to the Isles of Scilly model. The habitat suitability models all performed well within their own spatial realm, showing good fit within each case study site and for the whole UK and Ireland. However, transferability was more successful when the UK model here was applied to the smaller spatial extents of the case study sites rather than vice versa. So here the model shows much higher scores. On inspection of the broad scale model, it does appear to overpredict suitability around shallower coastline, as bathymetry and log PAR directly related to depth were found to be the most important variables in explaining seagrass presence overall. So why the differences? Well, Milford Haven, the Solent, Orkney and the Isles of Scilly are really complex systems. Other sites that were included in the RESO project, such as Porthincline and Studland Bay, are arguably more simple seagrass sites. Larger area sites, such as the Solent and Orkney, show better transferability to smaller sites. The models conducted at smaller spatial scales for the case study sites appear to be more sensible in determining suitable seagrass habitat when compared to the broad-scale British Isles model, which you can see in this diagram on the right-hand side. It is over-predicting where it is shallow, as you can see by the dark red patches, which indicate a suitability of closer to 1. On the left-hand side of that diagram, you can see that the site GLMs were predicting a bit more sensibly where seagrass is more likely to be able to be found. The smaller spatial habitat suitability models also did not transfer well to other case study sites, except for the Studland Bay and Orkney Isles GLM models. This is evident from the gradient of the localised conditions. The Orkney model may have performed better when applied to the Isles of Scilly model, as both sites are island groups, but the Isles of Scilly model did not transfer well to Orkney. Now, this is most likely due to the larger range of conditions in which seagrass areas are found around the Orkney Isles, with both deep and intertidal populations, and environmental ranges more similar to the UK ranges rather than the Isles of Scilly ranges. Seagrass meadows in the Isles of Scilly are mostly subtidal and more confined to sheltered and relatively shallow clear waters. Scale is therefore really important to consider for habitat suitability modelling. Spatially broader scale models will be able to capture the general large scale processes that drive population distributions, as seen with the larger broad scale British Isles, UK and Ireland model. And this will be able to explain distributions at smaller scales. However, fine scale models will be better at interpreting the local conditions, explaining local distributions, which would only be able to be extrapolated to areas with similar characteristics. Milford Haven shows the lowest transferability. This could be because it is a meadow that is in decline and where the conditions are no longer suitable. Analysis of monitoring data in the past have shown in the figure that's on the bottom left, these figures here, they show stability of more pristine seagrass meadows within the Isles of Scilly from monitoring data taken, showing shoot density and then wasting disease on the bottom. Whereas Little Wick Bay, which is found within Milford Haven, has shown declines in shoot density, but increases in wasting disease, which indicates poorer water quality. And then does that also indicate poorer suitability for seagrasses? Thank you for listening.
Hi everyone, and thank you for tuning in. I am Johannes Krause of the Institute of Environment at Florida International University. And today I will talk about some of our recent findings coming out of our seagrass and water quality monitoring programs in South Florida. My talk is titled, Seagrass Response to Nutrient Enrichment Depends on the Delivery Signal and Relative Nutrient Availability. And this work was published earlier this year in L&O. I would like to start with a brief overview of the seagrass monitoring program that is run by the Seagrass Ecosystems Research Lab at FIU with PI Jim Forkran. This program was first started in 1995 and then gradually expanded over the years. It was initially set up together with the water quality monitoring program to help monitor the water quality in the Florida Keys National Marine Sanctuary. In the late 1980s, there were seagrass die-offs occurring in the Florida Keys, and concerns about water quality prompted US Congress to establish the sanctuary. This map shows the monitoring sites in South Florida. The first set of 30 sites were established in 1995 along the Florida Keys, and half of the sites offshore, close to the Florida Reef Tract, and another half close to the shore, what we call nearshore sites. In 2012, 10 additional sites were established within a few hundred meters of the shore. This is called the halo zone. I will call these the inshore sites in some of the figures I show later. All of these sites are co-located with the water quality monitoring program that measures a wide array of water quality parameters in bottom and surface waters. The methodology we use is consistent with monitoring in Florida Bay. There are 5 sites from the Florida Coastal Everglades LTER, funded by the NSF, and then we also have another 23 sites in the Dry Tortugas National Park in the Gulf of Mexico, which we monitor together with the National Park Service. The monitoring design we use is based on fixed transects. At all of our sites, we installed permanent site markers with buoys. These sites are visited at least twice a year in the winter and summer for seagrass monitoring. At 10 pre-recorded random distances along the 50-meter monitoring transect, we assess the benthic community composition and cover using quadrats. We also collect sediment type data and measure the canopy height. Then in addition, we collect seagrass leaves of all species that are present at the sites for chemical analysis of tissue carbon, nitrogen, and phosphorus contents, stable isotope ratios, as well as morphometric measurements. Over the years, the data collected by this monitoring program produced a lot of insights into the ecology of seagrasses, especially with regards to nutrient dynamics in tropical carbonate systems. For example, this map here shows the spatial variability in seagrass tissue nutrient contents. Seagrass tissue nutrient content is a useful indicator of the nutrient regime and patterns of relative nutrient limitation, because seagrass tissues integrate the environmental signal over long periods of time. Depending on the species, seagrasses incorporate nitrogen and phosphorus at a specific ratio when both nutrients are present in abundance. This was termed the seagrass Redfield ratio, and for Thalassae testudinum, for example, it would be a molar ratio of N2P of about 27. Now, in phosphorus-limited environments, seagrasses will incorporate relatively more nitrogen into their tissues, and the N2P ratio will be higher than 27. In N-limited environments, seagrasses will incorporate relatively less nitrogen into their tissues, and the tissue entropy will be less than 27. In the map here, I show the striking gradients of nutrient limitation across the South Florida seascape. The warmer colors indicate higher seagrass tissue entropy or greater phosphorus limitation. Generally, seagrasses are more phosphorus limited in South Florida because phosphorus adsorbs to carbonate sediments and may be hard to access by primary producers. But phosphorus is delivered by ocean water, and at offshore locations, there can be enough phosphorus so that nitrogen becomes the limiting nutrient for seagrass growth. Phosphorus limitation is the most extreme near the coastline and in Florida Bay, where flushing is very restricted and nitrogen is delivered from runoff. This leads to regional differences in relative nutrient availability, with very phosphorus-limited regions in Florida Bay and inshore sites, while potentially more nitrogen-limited regions are the offshore and nearshore sites, as well as the central region in this map that we call the sluiceway. In addition to these spatial gradients, the long-term monitoring data also allowed us to look at temporal trends. And over the 30 or so years of water quality monitoring, there was this striking signal of interannual fluctuations in water total nitrogen to total phosphorus ratios. This is shown in this figure over here. We have water N to P on the y-axis and show time series by region, with the offshore, nearshore, inshore, sluiceway, and Florida Bay sites in different colors. A few observations about this signal. It is remarkably synchronized across sites and across regions, and appears pulsed with peaks and troughs in relative nutrient availability a few years apart. When looking for drivers of these trends in water quality, we considered climate and meteorological data. For example, we looked at regional precipitation and climate indices, such as Southern Oscillation Index, which measures the state of the El Niño Southern Oscillation. First, let's look a little bit more closely at precipitation. The left panel shows regional annual rainfall with Southern Oscillation Index. Negative Southern Oscillation Index corresponds to El Niño conditions, where South Florida experiences higher rainfall years. In the right panel, I show the relation of precipitation and water N2P ratios. In all regions, N2P is higher in high precipitation years, except for the inshore sites. There are a few different explanations for this pattern. It was recently shown that freshwater delivery to the shelf from South Florida rainfall can promote upwelling of nutrient-rich waters. Additionally, terrestrial runoff can have high concentrations of nitrogen that are transported to the coastal waters. Whatever the dominant process is, we found higher rainfall years were associated with higher nitrogen availability across South Florida. Okay, so to recap, in South Florida, we have temporal pulses of relative nutrient availability that meet a seascape with spatial gradients in relative nutrient limitation. Now, naturally, we want to know whether there are any effects on the seagrass systems. In the next few slides, I will show some of the different types of impacts of water quality that we could glean from our monitoring data. First, we found that seagrass cover actually showed fluctuations that were synchronous with water N2P ratios. At offshore, nearshore, and sluiceway sites, which are the more nitrogen-limited regions, peaks in seagrass cover are associated with periods of high nitrogen availability. However, in periods of low nitrogen availability, seagrass cover is reduced. Conversely, at the phosphorus-limited inshore and Florida Bay sites, peaks in seagrass cover are not synchronized with peaks of nitrogen availability. There seems to be some time lag. So there's evidence for impact of water quality on seagrass cover, but it differs by region. We can observe these relationships in a little more detail. Panel A of this figure shows that seagrass cover in the offshore, nearshore, and sluiceway regions increase with water N2P, while inshore and Florida Bay sites show the opposite trend. Similar relationships are apparent in panel B, this time with water total nitrogen concentrations. Panel C shows seagrass cover and seagrass tissue N2P and shows that more phosphorus-limited seagrasses generally have the lower cover. Together, these data suggest that increases in nitrogen concentrations which can cause eutrophication and seagrass degradation in other systems, is actually increasing seagrass cover in some regions of South Florida. Specifically, higher nitrogen concentrations can fertilize offshore, more nitrogen-limited meadows, while having no such effect on more phosphorus-limited seagrasses close to shore. From earlier work, we know that there are also other impacts of water quality on seagrass meadows, specifically at inshore sites that are within 500 meters of shore. At some of these sites, we found a continuous increase in phosphorus concentrations in the water column, likely due to leakage from septic tanks. We would consider this a press-type pattern of nutrient enrichment. Water phosphorus concentrations are shown in dashed blue lines in these graphs, with thalassia density in green and halodolae radii density in yellow. Here, we observed lasting changes in the seagrass species assemblages, switching from thalassia-dominated meadows to the fast-growing halodolae meadows as phosphorus concentration increases over time. So to wrap up, here are some takeaways. First, we found the situation in South Florida where pulses in nutrient availability impact seagrass meadows along gradients of nutrient limitation. From looking at seagrass cover, we found that nutrient enrichment affects seagrasses differently depending on their relative nutrient limitation. Some tropical seagrasses seem to be able to capitalize on increased nutrient availability if they were previously limited for that nutrient. This is the case at more offshore, back reef seagrass meadows in South Florida. This also shows that nitrogen fertilization from runoff affects meadows that are relatively far from the source, but can pass over phosphorus-limited meadows that are close to the source. In other words, proximity to the source is not proportional to the impact. Fourth, it is probably a characteristic of tropical carbonate environments that nitrogen concentrations can fluctuate, with nitrogen levels being reduced in years of low nitrogen delivery. Phosphorus, on the other hand, is better retained in the system and is leading to more permanent phosphorus enrichment. And lastly, it appears that disturbance type matters when looking at water quality impacts on seagrass meadows. The pulsed nitrogen enrichment led to periodic fertilization, but no lasting change in communities. In contrast, the press-type phosphorus enrichment we found at inshore sites led to permanent shifts in species composition. This is all that I have today, and I would like to thank you for listening in. I would also like to thank the Seagrass Monitoring PI, Jim Forkran, and everyone who worked in this program over the last 30 years. Feel free to reach out to me in the below email address, and make sure to check out our recent paper in L&O that I presented on today. I look forward to your questions in the Q&A.